Hey everyone, Mr. Pat here, and we have a very fun experiment today. So today, we'll be learning all about variation in plants, and we'll be turning into scientists by extracting DNA from these strawberries. So like always, everyone come gather in, put in your EcoSpaces thinking cap, because we are ready to dive right into these strawberries. From the feedback I got from everyone, I know that everyone wants to see strawberries grown, so today we're going to learn a lot about strawberries now. Now, what is it that makes all living things look the way they do? Those are called traits, and those are the physical characteristics we see in each other. Now, we all have different traits. We're the same human, but we all look different. We all have different hair color, different eye color, we're all different heights. Now, those are all traits. Now, if you look at this picture right there, how many different traits can you see in that picture of dogs? We see big ones, small ones, ones with more hair, longer hair, ones with no hair, very short hair, ones with bigger ears, ones with floppy ears, ones that point up. All different types of traits that we see in dogs, and the same thing goes for humans. So in essence, a trait is something that makes you, you. That makes sense. So when your mom or your dad or a guardian in your family says something like, you get the best traits from them, they're essentially saying you get that great, amazing smile from them, or that brilliant mind with a huge imagination. That's what they're saying. And usually all these traits are passed down from our parents generation to generation. And when it comes to variation in traits, those are things that's going to allow a plant or animal to survive. And when we learned about those desert plants just a few weeks ago, those all had specific traits that allowed them to survive those really harsh conditions. All right, so let's take a look at another picture so we can see even more difference in traits. So I'll whip up some apples. Perfect. So can anyone tell me the differences they see right away? Size, yes, they're all different sizes, all different shapes. Color, yes, as we can see, all different colors or even slightly different. And there's one last one that comes to my mind, but it may not be as obvious. Someone just said it. Say it again one more time. There it is. Taste. They are all going to taste very, very different. Now let's take, for example, the Red Delicious apple and the Granny Smith apple. A Granny Smith tree is always going to produce Granny Smith apples, and a Red Delicious tree is always going to produce Red Delicious apples. Now, how? So all cells in living plants, animals, and even bacteria have this special chemical substance that holds these traits so they pass on to the next generation. And that is why we see Granny Smith trees only producing Granny Smith apples, because those seeds in any type of plant will only produce that type of plant. And you're thinking, what is the name of this chemical substance? And you all probably may have heard of it before, but it's super simple. It is what we call DNA. And like I mentioned earlier, we'll be breaking down our strawberries to extract DNA. Now, strawberries are perfect for extracting DNA because not only are they easy to break down, but they also contain these enzymes called pectinases and cellulases that help break down the cell walls. And most importantly, strawberries contain 8 copies of each chromosome, so they have a lot of DNA to extract. So let's take a look at all the supplies we will need for our experiment. So obviously we will need our strawberries, a plastic bag, a coffee filter, plastic cup, and we also will be using half a cup of water, 2 teaspoons of dish detergent, and this is going to help dissolve the cell membrane, which is a lipid bilayer. 1 teaspoon of salt, and this is going to help remove the proteins that are bound to the DNA. We'll also be using rubbing alcohol, and about a half a cup of this, and this will cause the DNA to precipitate. And when the DNA comes out of the solution, it tends to clump together really, really tight, and it makes it super visible. And the long strands of DNA will wrap around this coffee stirrer so we can see and play around with it. Now to prepare, I'm going to put the rubbing alcohol in a freezer just for a little bit as I prepare the rest of the experiment. First things first, gotta take off all the green leaves. Perfect. Now, put the strawberries in the bag, close it up. We'll be gently smashing for about 2 minutes. Alright, we completely broke down the strawberry, which is great because now we broke down those cell walls and now all that DNA is being released. Now it's time to make our extraction liquid. And essentially, extraction is basically separating two substances that are mixed together. So in this cup, we'll be mixing 2 teaspoons of dish detergent, 1 teaspoon of salt, and then a half a cup of water to make our extraction liquid. So I got here my DNA extraction liquid, and I'm gonna add 2 teaspoons into the bag, reseal it, and then smash again for another minute or so. And this is gonna furthermore break down the cells and extract more DNA. And like I mentioned, that salt is gonna help all that DNA clump together so we could all see it really clearly. Here we go, 2 teaspoons back into the bag. That's 1 teaspoon. And 2. And then smash it again and we'll be right back. So now that it's all broken down again, I'm gonna take my coffee filter and cup, put the coffee filter in the cup, take my strawberry, and nicely pour it in the cup, just like that. Get it all. Perfect. Set that aside and we'll let it drain and tie this at the top so we can squeeze out just like this. Y'all see all that coming out? Perfect. Does get a little messy. But after a few minutes, we should be good to go. And here's our final product. So now, let's go all the way to the freezer. Grab our isopropyl rubbing alcohol. Come all the way back. And we're gonna measure out an amount that is the exact same height as that liquid. So now we will slowly pour the alcohol down the side of this cup, and we're not gonna stir or mix or blend or anything. Because right now, we have just isolated the DNA. And look, y'all, you see that cloudy substance? That is all the DNA we were able to extract from just 2 strawberries. Like, isn't that so cool? And I did it on my first try. I was surprised. I thought I had to do it over and over again. Here, I'm gonna take my stir. Yeah, look at all that DNA. Crazy. All right, y'all, that is it for the experiment, and I'm honestly super happy that it worked out the first time around. I was getting worried. Usually it should happen in a few seconds, but I waited like 2 minutes, and then I saw all that DNA clumped up together, and all because of that salt. So amazing. So I hope everyone had a really fun time learning about how to extract DNA from a strawberry. And if you did the experiment with me, and you got some leftover strawberries, go right ahead and dig in. But I'll see y'all next time for one of our last lessons. Sad, but I'm looking forward to it because we have a very fun final project after. But I will see y'all at the next lesson.